Hello, my name is Angelina and I will be doing my local expert project on 3D cell cultures. Let's start with what 3D cell culture is. It is a type of cell culture, like we've discussed in class, and it is a 3D culture environment that allows cells to interact with each other and the extracellular framework they're growing in. There are two types of 3D cell cultures, with scaffolding and without scaffolding. Some popular types of scaffolding methods include hydrogels and inert matrices. Some popular types of non-scaffolding methods include low adhesion plates, micro-patterned surfaces, and hanging drops. You can use these methods to form organoids, which is when cells differentiate and self-assemble into things called organoids where the native environment of the original tissue or organ is mimicked. Microfluidic organ-on-a-chip models can also be used, and this is where a 3D microfluidic chip is engineered to mimic the physiology of an organ. Channels in the chip allow the flow of liquid throughout the culture to deliver nutrients. Although this technique is showing a lot of promise, there are some limitations to it. First, it requires highly skilled personnel and strict aseptic technique to perform. Also, animal and human cell cultures grow very slow compared to microorganisms like bacteria and fungi. This technique and its components can also be very expensive and time-consuming for potentially not a lot of cells to be made. Reproducibility can also be difficult because of the variability in the cell cultures. Additionally, measurements and microscopic analysis can be difficult because of the larger size of some of these 3D cell cultures. Finally, it can be challenging to deliver the proper nutrients to some of the cells because of the density and size of these cultures. Even though there are some limitations for 3D cell culturing, there are also a lot of advantages. First, it is more physiologically relevant and predictive and better represents in vivo tissue because cells in natural environments grow and interact with other cells and this impacts their function. This environment is better represented in 3D cell cultures compared to 2D. It also better represents human tissue outside of the body in terms of growth and how they respond to disease and injury. 3D cells have higher structural complexity and can maintain homeostasis for longer. These cultures also better portray the integration of flow for things like blood, interstitial fluid, and urine flow, and the barrier tissues that separate organ compartments. Additionally, 3D cell cultures decrease the use of animal models as it can be unethical and animal models don't always accurately represent the targeted organism. Finally, the expression of genes and the biochemistry of cells are more accurately represented in these 3D cell cultures. Most of the controls for 3D cell culturing depends on what type of cell culturing is being done. The controls depend on the method of 3D cell culturing being used, the types of cells being used, and how they need to be treated, and the types of environment the cells need to grow in. Some basic controls for this type of cell culturing could include having cells that aren't getting treated with what's being tested, the media is the same for all of the cells, and all of the cells get the same cultivation treatment. This figure is from the paper Comparison of 2D and 3D Culture Models as Drug Testing Platforms in Breast Cancer. In this paper, it was found that 3D cell cultures were more likely to represent the in vivo desired conditions compared to 2D cell cultures. In this figure, it is shown that the breast cancer cells form dense multicellular spheroids in the 3D cell cultures, and this decreased the cancer cell's sensitivity to chemotherapeutic drugs. In the graphs on the very right, you can see that the drug sensitivity for the 3D cultures was decreased compared to the 2D cultures. This figure is from the paper Altered Ubiquitin Signaling Induces Alzheimer's Disease-like Hallmarks in a 3-Dimension Human Neural Cell Culture Model. In this paper, they used 3D cell cultures to grow their neuronal cultures. Specifically, they used a matrigel, which is a hydrogel used for scaffolding growth of the cell cultures. It was found that the 3D culturing allowed for a more realistic growth of the neuronal cultures and how cells reacted in the disease state. This figure is from the paper The Impact of the Cellular Environment in Aging Modeling Alzheimer's Disease in 3D Cell Culture Models. In this paper, they used 3D cell culture to try and mimic the environment in the brains of patients with Alzheimer's disease. They found that the 3D cell cultures were able to better represent the amyloid beta plaques found in Alzheimer's disease brains compared to 2D cultures. Additionally, they found that the 3D extracellular matrix can better mediate disease pathogenesis. These are the sources I used if you wanted to learn more about 3D cell culturing. Thank you for watching my presentation on 3D cell culturing, and let me know if you have any questions.